This is the lecture for Chapter 18, Practical Applications of Immunology, Part 2. All the practical applications that we talk about in this chapter have to do with the serum proteins, the antibodies. And again, antibodies are proteins, and they're able to recognize the epitope of different antigens. So they do that using the variable region of the heavy and light chain. It has a particular shape that fits perfectly the epitope of a particular antigen. In Part 1, I talked about how antibodies can be used for different types of therapies, such as vaccination, anti-venom, antitoxin, and IVIG. In this part of the lecture, I'm going to start talking about diagnostics. Diagnostics are used to determine if an individual has a particular disease. And for this class, we are going to focus on infectious diseases. So for the diagnostic tests that we're talking about, they are used to indicate if a person has an infection. That means if they have been invaded by the microbe, whether or not they are expressing signs and symptoms. It can also determine what sort of disease, infectious disease, a person has. So if they have a disease, they are expressing signs and symptoms. And then these tests can also determine if a person is immune. If they were infected with a microbe and that microbe stimulated a primary response, and now they have antibodies against that particular microbe. So for all of these tests, you are indicating whether the person has the actual pathogen, or you're looking for evidence of antibodies which indicate the person came in contact with that particular pathogen. In some cases, some of these tests can be used to test for other molecules. And these other molecules have to be some sort of antigen, which means that they have to be some sort of large protein. For all of these tests, when you are designing a test, you need to make sure it has a high level of sensitivity. And sensitivity is the ability of the test to determine or identify a positive sample. Also, these tests need to have a high level of specificity. And specificity is the ability for the test to not react with a negative sample. When developing a diagnostic test for an infectious disease, you can look for the evidence of two different things. You can either look for evidence of the antigen, which indicates the actual presence of the microbe, or you can look for the antibodies, which indicate that the individual was exposed to the microbe and has generated an immune response. Also, specificity is really important because often you want to know exactly what strain of the microbe has infected the individual. So flu strains, you may have heard different names for different flu strains. One is H1N1, and the H and the N indicate different antigens that you find on the surface of the virus. So there's H1N1, and then there's H2N2, H3N3. So there are all sorts of different antigens that you find on the surface. And to identify the particular strain of flu that is infecting a person, you want to identify either the presence of that exact antigen or antibodies that recognize that antigen. So this may be the antigen involved with N1, and this virus could be H1N2. And this could be the 2 antigen, and these would be the antibodies that only recognize the 2 antigen. And so that would be two different ways you can identify different strains of flu virus, either looking for the exact antigen on the flu virus or looking for the person's antibodies that would recognize the different strains of the flu virus. When designing a diagnostic test, you have to keep in mind what are you trying to identify. So in terms of an infectious disease, you are either trying to identify presence of the antigen or presence of the antibody. So in designing a test, you have to figure out which one do you want to identify. If you want to identify presence of the antigen, then the test must include the antibody, which will be used to identify the antigen. If instead you want to identify the presence of the antibody, for instance, you want to determine if a person is immune to a particular disease, then you need to have the antigen in the test. So all of these tests I'm going to talk about involve antibodies binding to antigen. But you have to keep in mind, what is the test detecting? Is it detecting the antigen? Then that means the antibody has to be in the test kit. Or is it determining the presence of the antibody? If so, then the antigen has to be in the test kit. For most of these tests, I will be talking about identifying antigen. So in the test kit, you have to produce antibodies that will recognize the antigen in the patient's sample. If a test requires antibodies to identify the antigen in a patient sample, then you need to have those antibodies produced. And the easiest way to produce antibodies is to stimulate a primary response in an animal. So remember, before in the lecture I talked about the primary response. Primary response, you have mature B cells in the lymph nodes waiting to be stimulated by an antigen. So if you inject a particular antigen into an individual, and that individual has a B cell that will recognize that antigen, then that B cell goes through clonal expansion, generating memory cells and plasma cells. And those plasma cells are going to secrete antibody. So one way to generate antibodies that can be used in a diagnostic test is to take a test animal. Usually a rabbit is what they have used, a rabbit. They inject the rabbit with the antigen they are looking for. So let's say it's H1N1. You inject the N1 antigen from the flu virus into the rabbit. If the rabbit has an B cell that responds to the N1 antigen, then it will go through mitosis, clonal expansion, and all the antibodies that are generated will recognize that N1 antigen. So then all you have to do is take blood from that rabbit, purify the antibodies, and now you have antibodies you can use in a diagnostic test. The one problem with using animals to generate antibodies to a particular antigen is that often they will end up being polyclonal. Polyclonal, poly means many, many clones, many B cell clones are responding. So what happens is that actually that N1 antigen, which is a spike found in the enveloped virus, so this is the envelope, this is the spike in the virus, that antigen N1 will have several epitopes. This could be epitope 1, this could be epitope 2, and this area could be epitope 3. Remember, every antigen has several epitopes. So what could happen is that this B cell could be responding to epitope 1. This B cell may be responding to epitope 2, and this B cell may be responding to epitope 3. So you have 3 clones in this example producing antibodies to this particular antigen. So that's polyclonal antibodies. So what that means is that all those antibodies are not identical. Some recognize epitope 1, some recognize epitope 2, some recognize epitope 3. And the problem comes in if the other antigen, N2, if it has a similar epitope. What if it shares epitope 3 with N1? So if you have polyclonal antibodies in your diagnostic test, you may get cross-reactivity. You may get positive reactions for N1, which you're looking for, but you may also get positive reactions for N2. So then you do not have high specificity in your diagnostic test because it is not only identifying N1, it is cross-reacting with another strain of flu virus. To prevent the cross-reactivity that can happen with polyclonal antibodies, scientists have developed a method to generate antibodies from one B cell clone, and they call these monoclonal antibodies, or MAB. Monoclonal antibodies are much better than polyclonal because they are uniform. They come from one B cell clone, so they identify exactly the same epitope on a particular antigen. Also, because of this, they are highly specific. So they are very specific, and they will only identify that one particular antigen because they bind to that one particular epitope. And the other thing about monoclonal antibodies is that you can produce antibodies in large quantities. So this is a very efficient and relatively cheap way to generate highly specific uniform antibodies in large quantities, so you can get highly specific uniform diagnostic tests. To generate monoclonal antibodies, the first thing you do is you take your antigen of interest. So it could be the antigen for the flu virus, it could be the N1 antigen, or if you were developing a test for the coronavirus, you would take one of the spike proteins from the enveloped coronavirus and inject it into the mouse. Then the mouse is going to go through a primary response. So you inject the antigen into the mouse's B cells get stimulated, activated, they go through clonal expansion, generating plasma cells which create the proteins, and those plasma cells will be found in the spleen or the lymph nodes of the mouse. So it's those B cells, those plasma cells that you want to get, so you will have to remove the spleen and the lymph nodes from the mouse and take out the B cells. Now, B cells by themselves will not survive very long in culture. They will only survive for about 2 weeks in culture, and you want a constant source of antibodies for your diagnostic tests. So what scientists do is they mix the B cells from the mouse with cancerous B cells. And cancer cells, you know, cancer are cells that go through mitosis constantly. So they basically can live in culture indefinitely, forever. Sometimes they're called immortalized cells. When you mix the cancerous B cells with the B cells from the mouse, some of those cells will fuse together and form hybrid cells. So those hybrid cells are cells that produce antibodies, but you can maintain them in culture indefinitely, almost forever. Once you have mixed the spleen cells with the cancer cells and created some hybrid cells, then you separate those cells into individual wells. So every cell has its own well. And once you do that, you identify the hybridomas. So those B cells that have fused with cancer cells and can live indefinitely. Once you identify the hybridomas, then you identify the one that is producing the antibody you want. So this is the hybrid cell that you can maintain in culture, basically forever, that is producing antibodies that recognize the antigen that you injected into the mouse. So to produce monoclonal antibodies, you have to have the protein, the antigen that you want to identify. You inject that antigen into the mouse. The mouse generates antibody-producing B cells. You mix those B cells with cancerous cells to form hybrid cells. Once you have formed those hybridomas, you sort them and screen them to find the one that is producing the antibody that recognizes the antigen you injected in the first place. And again, once you have done this, it is very easy to maintain these cells in culture, and now you have a constant source of the antibody you can use for all your diagnostic tests. I am going to finish this lecture by going over two different types of tests that use monoclonal antibodies. So the first type of test is precipitation reaction. Precipitation reactions involve using IgG, so this is probably IgG. Remember, IgG is a monomer, or IgM, so IgG or IgM. IgM, remember, is a pentamer, and it's using those antibodies to identify soluble antigen, so antigen floating around in a solution. And a precipitation reaction is using the result of agglutination. So the ability of antibodies to bind to antigen and to clump them together. For a precipitation reaction, it is concentration dependent. So you need to get to a particular concentration where you have a zone of equivalence, where the concentration of antibody and antigen is in balance and you get these clumps forming. If you have too much antibody, you won't get the clumping. If you have too much antigen, you also won't get the clumping. So you have to have this particular ratio of antigen to antibody, and that is called the zone of equivalence, where you actually see the reaction. In a precipitation reaction, this should remind you of chemistry. When you made precipitates, you take two clear fluids, mix them together, and you form solid particles. That's what will happen in a precipitation reaction. So an example of a precipitation reaction, this is a cartoon. You start out with a test tube, and in the test tube you have a solution that contains the antibodies. Then you add the patient's sample, could be a blood sample where you have those potential antigens, and as those antigens diffuse into the test tube, you will reach a zone of equivalence where you will see a white precipitate form. So this is an actual example. So the antibodies were in the solution here. You added some patient serum there that has potentially the antigen. The antigen diffused down into the antibodies, and where you see this cloudy white precipitate forming, that is the precipitation band. So that is a positive reaction. So whatever antigen you were looking for in the patient's serum, if it's a virus, if it's a hormone, if it's a drug, whatever protein you were looking for in the patient's serum, this patient is positive because you have that white cloudy precipitate forming. Precipitation reactions can also occur in solids, in agar, and in this case you can incorporate the antibody into the agar. So the antibody is found in the agar, and then you drop a drop of the patient's sample onto the agar, and the patient's sample may have the antigen. And as the patient's sample diffuses through the agar from the drop, when you get to the right concentration, you will get a white precipitate forming. And an actual example of this, here you have agar that has antibody in it. You put a drop of antigen here, and as the antigen diffuses through the agar, you get a precipitate forming, so that is a positive reaction. Agglutination tests are very similar to precipitation reactions because it's taking advantage, of course, of the result of antibody binding to antigen. which is agglutination. But the difference for the agglutination test is that you are not looking for soluble antigen, you are looking for antigens on large particles, such as cells. So agglutination reactions are still going to involve IgG or IgM, but this time the antigens are actually on large objects. They are not soluble antigens. This is an example of a direct agglutination test. A direct agglutination test involves the antibody, in this case IgM, because it's a pentamer, IgM identifying antigen on the surface of a bacterial cell. So this agglutination test is not identifying soluble antigen, but identifying antigen that is still bound to a bacterial cell. So this is a direct agglutination test. The most common use for direct agglutination tests is with blood typing. So blood typing, you should know that you have different blood types, A type, B type, AB, O type, and that all involves antigen on the red blood cells. So B type blood has B antigen on the red blood cells, and so to identify the presence of B antigen on the red blood cells, you can use IgG that is specific for the B antigen. When determining a person's blood type, you would test their blood using 3 different types of antibodies. So one type of antibody is anti-A, it identifies the A antigen. The other is anti-B, which refers to identifying the B antigen, and then anti-D is identifying the Rh factor. And the Rh factor determines if a person has a positive or negative type of blood. So to perform a blood test, you would take a drop of blood, put it into a well or onto a paper, and you would add a drop of antibody that recognizes antigen A. If the red blood cells have the A antigen on them, they will agglutinate. They will clump together and you'll see that particulate look. To test for B antigen, you add a drop of blood, add a drop of antibody that recognizes the B antigen, and here you can see there is no agglutination. It still remains a general color, so this is negative. So this is a positive reaction, this is negative. You would also test for the Rh factor. You would add a drop of blood, add a drop of antibody against the Rh factor, and here you get agglutination, so this is a positive reaction. So this person's blood type is A positive because they are positive for the A antigen, positive for the Rh factor, negative for the B antigen. And this is an example of a direct agglutination reaction. Indirect agglutination reactions are still agglutination reactions because they are looking for antigen that is not soluble. So it's bound to a larger structure. And for an indirect agglutination reaction, the antigens are found on latex beads. So latex beads, they're very, very small plastic beads. And in this case, you generate the latex beads in the test. So you put the antigen on the beads, and then you use these beads to identify the presence of the antibody in the patient's sample. So this could be used to identify if an individual has produced antibodies to that particular antigen. Indirect agglutination reactions can also be used to identify the presence of the microbe in a sample. So in this case, again, indirect agglutination reactions, you're using the latex beads. This time, the latex beads have the antibodies bound to them. So those monoclonal antibodies that you generated against that particular antigen. So you can use these latex beads to identify the presence of a bacterium in this example in a sample. And this can be used to identify the presence of E. coli in water. So if you have contaminated water, you take the latex beads that identify the particular strain of E. coli, you add a sample of water, and if you get agglutination, then that water is positive for presence of E. coli. This is an example of an indirect agglutination test for the presence of Staph aureus. So you are looking for the presence of Staph aureus in different samples. So therefore, on the latex beads, you have rabbit IgG that is specific for the Staph aureus. So you take a sample of your patient's sample, you add a drop of the latex particles, and then if you see agglutination, that means that sample is positive for the Staph aureus. So it looks like sample 1, patient 1, sample 1 is positive, sample 2 is positive, 3 is positive, 4 is positive, 5 is positive, and 6 is negative. So again, this is an indirect agglutination test, and this one is used to identify the presence of an antigen on a particular microbe, which is helping you identify that microbe.